Welcome to the Deep Dive, where we take your stack of sources and pull out the most important nuggets of knowledge and insight.
Today, we are going way back to the very language that life uses to write itself, the foundational rules of inheritance.
We're diving into Mendelian inheritance,
Yeah.
and it all starts with something really simple, a characteristic you might even have yourself. Dimples.
Dimples, yeah, or a cleft chin, a widow's peak. These little markers are all characteristics encoded right there in your DNA.
They're genetic traits.
Exactly. And when they follow a really simple, predictable pattern, controlled by a single gene with just two versions, two alleles, we call that simple, or Mendelian inheritance.
It sounds like Biology 101, but the implications are just massive. So our goal today is sort of threefold. We're going to quickly review the foundational rules that Gregor Mendel established. Then we'll get into the practical tools, Punnett squares and pedigrees, that let you predict and track these traits.
And then the really interesting part.
Yes. The real mission here is to explore why this simple system is often, well, a bit misleading when you look at it through the lens of modern genomics.
Exactly. It's like we're using Mendel as the essential grammar. But we have to remember that the real world and real genetics rarely speaks in such simple sentences.
Okay, so let's start at the beginning with the revolutionary idea itself. We have to go back to the Austrian monk, Gregor Mendel, in his monastery garden. This is the mid-19th century.
Right. And he wasn't a geneticist like we think of one today. He was really a mathematician who saw patterns. He was just meticulously tracking traits in thousands and thousands of pea plants.
Things like their height or seed color.
Yeah, and his key insight was that traits weren't blended. That was the thinking at the time. That offspring were just a physical mix of their parents, like mixing two cans of paint.
Right.
Mendel showed that no, heredity was controlled by discrete little factors, what we now call genes.
And when we look at these simple traits today, we group them based on where those genes actually live in our cells. There are two big categories, right?
What we do. The first and most common are autosomal traits. These are controlled by genes on the 22 pairs of autosomes.
So everything that isn't a sex chromosome.
Exactly. And since males and females have the same set of autosomes, the inheritance pattern looks pretty much the same for both.
But then things get interesting when we move over to the sex chromosomes.
Absolutely. Those are the sex-linked traits, and they're generally controlled by genes on the X chromosome. The Y chromosome is it's so small it has very few genes, so the patterns for these X-linked traits become really distinct.
So what were the two elegant core rules that Mendel pulled out of all these patterns?
The first one is the law of segregation. I mean, you can think of it as the ultimate fairness rule for making gametes.
Okay.
Every organism has two alleles for a gene, right? But when they make their reproductive cell, sperm or egg, those two alleles have to separate. They have to segregate.
So that each sperm or egg only gets one of the two options.
Only one. And this isn't an abstract idea. This is biology physically pulling those options apart during meiosis, specifically in anaphase I.
And the second rule?
The second is the law of independent assortment. This rule suggests that inheriting one gene is completely independent of inheriting any other gene.
So if a pea plant inherits the allele for yellow seeds, that has zero influence on whether it gets the allele for being tall or short.
That was the idea. But that's the one that comes with a big modern asterisk next to
A
it.
huge caveat, right?
Yeah.
So what's the refinement science had to make to that law?
Well, it's not universally true because of recombination or crossing over. During prophase I of meiosis, chromosomes can literally swap segments. So if two genes are located really close together on the same chromosome, they're likely to be inherited together. They're linked. They do not assort independently.
So Mendel just got lucky. Or was he a genius for picking the right traits?
A bit of both, probably. He chose traits whose genes were either on totally separate chromosomes or just very far apart, which let him see that simple, elegant rule without the complication.
That simplification let him move forward. Okay, so let's move from the theory to prediction. If you want to figure out what traits might show up in the next generation, you need two really practical tools, the Punnett square and the pedigree.
The Punnett square is our forward-looking tool. It's just a simple chart that helps us visualize every possible combination of alleles from the parents.
And it lets us figure out the expected ratio of genotypes in the kids.
Precisely.
Let's take that classic monohybrid cross-A, two heterozygous parents, big B, little b. You put the gametes on the axes, fill in the four squares, But the real insight here isn't just the diagram, it's the probability it reveals.
Exactly. The result is a 1 to 2 to 1 genotype ratio, 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive.
But what you actually see, the phenotype, is driven by that dominant allele.
Right. So the probability of seeing the dominant trait is 75%, or 3 out of 4. What this really shows is that genetics is fundamentally a game of chance. It's all based on which allele happens to end up in which gamete.
And if we scale this up to track two traits at once, the dihybrid cross. Let's say we're crossing two plants that are heterozygous for both yellow and round peas. The Punnett square explodes to 16 squares.
It gets complex fast, but that 16-square grid is the perfect illustration of the law of independent assortment in action. The 4 possible gametes from one parent combine randomly with the 4 from the other, and you always get that resulting phenotypic ratio of 9 to 3 to 3 to 1.
That's where you see the 9/16 having both dominant traits, round and yellow, and that lone 1/16 at the end with both recessive traits, wrinkled and green.
It's a beautiful mathematical confirmation of how it all works.
Okay, so the Punnett square helps us predict what's coming next, but also we need to look backward to map what's already happened. And that's the job of the pedigree.
A pedigree chart is basically a standardized family tree. It's designed to trace how one specific trait moves through generations.
And there are standard symbols, right? Squares for males, circles for females.
Yep. And we shade in the individuals who are affected by whatever trait we're tracking. But the real power is in reading the pattern. You can often tell the mode of inheritance just by looking at it.
So what are the quick visual checks? How do you tell an autosomal dominant trait from a recessive one?
For an autosomal dominant trait, you look for one thing. Every single affected person will have at least one affected parent. It very rarely skips a generation. It's relentless.
Okay, and recessive.
Autosomal recessive is different. You'll often see it skip generations. And here's the key. You can have 2 totally unaffected parents who are heterozygous carriers.
Have an affected child.
Exactly. That pattern where the trait just appears seemingly out of nowhere, that immediately screams recessive.
Let's shift this over to us, to humans, and see how these rules play out. We mentioned widow's peak and freckles as common examples of autosomal dominant traits.
Right. If we use, say, big W for the dominant widow's peak allele, it doesn't matter if your genotype is big W, or big W, little w, you get the trait.
And before we get to the sex-length stuff, it's probably worth touching on how sex itself is inherited.
A quick but crucial point. The father is the sole determinant of the baby's sex.
Because mothers only have X chromosomes to give.
Right. The father can pass on either an X or a Y, so it's a pure 50-50 coin flip for every single conception.
And that 50-50 ratio has a huge impact on the likelihood of X-linked recessive traits, which are famously more common in males.
Yeah.
Things like red-green colorblindness.
That's because males are operating with only one X chromosome, which they always get from their mother. If that single X carries the recessive allele for colorblindness, that's it. There's no dominant allele on the Y chromosome to mask it.
So they express the trait immediately.
Immediately. Whereas females have this sort of built-in safeguard.
The second X chromosome.
Exactly. A female has 2 X's. If she inherits one recessive colorblindness allele and one dominant normal one, the dominant allele masks the recessive one. She becomes a carrier.
She can pass the trait on, but she isn't colorblind herself.
Right. For a female to actually be colorblind, she has to inherit that recessive allele from both of her parents, which is just statistically far more rare.
And the real-world impact of colorblindness is much bigger than just struggling with that Ishihara test, the colored dots with the number hidden inside.
Oh, the practical dangers are significant. Think about jobs where color is critical. Pilots, electricians, police officers. A simple mistake with a color-coded wire or a map could have huge consequences.
Even just cooking.
Yeah, even cooking.
Yeah.
It can be hard to tell if meat is safely cooked if you can't distinguish the shades of red and brown. These simple Mendelian rules have really profound effects on everyday life.
And we can't really talk about X-linked inheritance without telling the story of hemophilia.
Ah, yes, the royal disease.
The royal disease.
Hemophilia is a recessive X-linked blood clotting disorder, and it got that nickname because of how it spread through the royal houses of Europe in the 19th and 20th century.
And it all traces back to Queen Victoria.
She was the key figure. She was a carrier. She didn't suffer from it herself, but she passed that allele to several of her daughters, who then married into the royal families of Russia, Spain, and Germany. And her son, Prince Leopold, he suffered from it directly. It's just a powerful, tragic example of how simple genetics can shape human history.
So we've laid it all out. Simple inheritance, predictable math, clear patterns. But now we get to the modern deep dive, where we have to ask, is this simple model actually true for most of life? This is where we leave the p-plans behind.
And the simple answer is no, not really. Mendelian simplicity is profoundly deceptive.
Deceptive.
Mendel's success was based on studying traits that had a sharp and certain separation, tall or short, yellow or green, no messy in between. But the vast majority of traits in humans, height, weight, intelligence, even hair color.
They're not on-off switches, they're on a spectrum.
Exactly. They are complex genetic traits. They are not controlled by one gene. We're talking dozens, sometimes hundreds of genes, all modulated by the environment.
This helps us debunk that phrase you always hear, the gene for.
Yes. That idea of the gene for eye color or the gene for tongue rolling. These textbook examples are often hugely oversimplified, or we now know they're polygenic. They involve many genes working together.
So genes don't determine the trait.
They don't. They shape the landscape of probabilities.
I think we need a better analogy here. If Mendelian inheritance is a simple light switch on or off, what's polygenic inheritance?
It's more like a massive, sophisticated mixing board in a recording studio. You've got hundreds of sliders, and each slider, each gene, contributes just a little bit to the final sound, or in this case, the final trait, like your height. Some genes push it up a bit, others push it down a bit.
And your final height is the sum of all those tiny effects.
The sum of all those small interacting effects. And we can't forget the environment, which is the other massive factor the simple Mendelian model just ignores.
It's like the person controlling the mixing board.
That's a great way to put it. The environment is the external control. Genes give you the potential for a certain height, but environmental factors like nutrition in childhood or exposure to disease can raise or lower that potential.
I think the diabetes example is a really good one.
It is. Carrying a genetic risk factor for type 2 diabetes doesn't seal your fate. Those genes might load the gun, but your lifestyle choices, your diet and exercise can fundamentally shift the probability of the disease ever happening. They can keep the trigger from being pulled.
So Mendel gave us the essential language, but modern genomics shows us it's a conversation that's way more intricate than just two alleles fighting for dominance.
It is. The whole field has shifted from deterministic certainty to probabilistic prediction. It reflects this multi-gene, multi-flinger reality.
And even the advanced math models that geneticists use today struggle with this.
They do. These models, they try to generalize Mendel's rules for huge populations, but they struggle precisely because they have to account for all these complex interaction effects. That simple 3-to-1 ratio that made Mendel famous. It almost never holds up when you have dozens of genes and environmental factors all in the mix at once. It's a testament to the sheer complexity of biology.
So we started with a simple P-cross. We learned how to track royal diseases. And we've ended up in a world where genes only really offer a probability, not a certainty. Mendel gave us the foundation for prediction, but prediction just isn't the same as a guaranteed outcome.
Precisely, and that, I think, leads to a final thought for you to chew on.
Okay.
When you do that simple cross of two heterozygous plants for flower color, the math predicts 75% will be purple. But if you actually go and plant 100 of those seeds and you get, say, 59 purple and 41 white, that result is still statistically possible.
How is that possible if the math is so clean?
Because probability only dictates the likelihood in the long run, in the aggregate. The chance of getting a recessive white flower is 25% every single time, for every single seed.
The outcome of the last seed doesn't change the odds for the next one.
Not at all. It's like flipping a coin. True certainty, where the final outcome matches the prediction exactly.
Yeah.
That's exceedingly rare in the beautiful random roll of the genetic dice.